Good afternoon, everyone. Thank you for joining us today for MBF Bioscience's webinar, Optimization Techniques for Accurate Quantitative Analysis of C. elegans Behavior. I'm excited to have joining us later during our Q&A session, the original developer of WormLab, Dr. Nicolas Roussel, Senior Research Scientist here at MBF Bioscience. My name is Peter Lang, and I am Product Manager for WormLab. One of my primary responsibilities here is to facilitate communication between the worm research community and our development team as part of a continuous improvement process for WormLab. So this is really a great opportunity to share experiences and feedback together. C. elegans is a very useful model organism, widely used in genetic, neurological, and developmental biology research applications, and exhibits a range of behaviors that can be quantitatively analyzed to gain insights into underlying biological processes. Visualizing these tiny transparent worms and accurately tracking through complex movements like coiling, entanglements, or investigating effects on social interactions presents significant challenges. WormLab is up to those challenges. Today we're going to give you an overview of WormLab, discuss some parameter optimization techniques to enhance the accuracy of your behavioral studies, and review some of WormLab's comprehensive analysis tools, followed by a Q&A session. A couple of logistical matters we need to get out of the way first. Feel free to ask any questions during the webinar using the Q&A button near the bottom of the Zoom window. This will open the question and answer panel. Simply type your question, press enter, and it will be submitted to us. All right, I think we're ready to get started. So let's go with the overview of the WormLab software. All right, so here we go. This is the WormLab software. Maximize that here. This is our user interface. So as you can see, we've tried to keep it very simple and user-friendly. In the upper left here, we have our menu items, and then our two main workflows, which we're going to go over today, namely the track workflow and the batch workflow. So today we're going to start with this track workflow. The first thing that we want to do, going along the steps here, is to import an image sequence. So I'll click Import Image Sequence, browse to the file, open, and as you can see here, we have our sample video. The sample video is about 15 seconds long, around 359 frames. It was acquired using our WormLab imaging system. This video exhibits good resolution, as you can see if we zoom in here, high contrast between the worms and the background, but there still is some illumination variation, debris in the background. This is typical of a worm plate image with our system. It will serve as a realistic example for our demonstration purposes today. All right, so once we have our image imported, we'll go on to the next step, which is actually setting the sequence info. So here on the right-hand side, you'll see the tools associated with setting the sequence info. Also want to describe some of the video playback tools that we have here along the bottom of the screen that allow us to actually navigate the video itself. So along the bottom here, we have our play button, which allows us to play through the video. You can zoom in or zoom out using mouse wheel control, jump to the end of the video, to the beginning, move frame by frame, or you could use a convenient scroll bar to actually drag through the timeline of the video. We have our frame number here on the left and our playback speed here on the right. On the right side of the window here in our sequence info, it tells us all the information regarding the resolution, the captured frame rate. And here's kind of the important part of this particular step is actually the scaling information. So WormLab does read a lot of information from the metadata from the video files themselves, like we see this one here. However, typically what's not included in that data is actual scaling information. So in order to actually utilize the metrics that are generated by WormLab that we'll see a little later, we need to define a micron to pixel scaling associated with the video so that the data that we are displaying in our analysis section actually is in a real world unit of measure. So what we need to do is define this micron to pixel scaling. Now, the way you would normally do this is with your acquisition system set up, you'd actually, like we have here with our WormLab system, we would take an image of a calibration standard. So in this case, I'll show you, we took an image of this millimeter scale with the system. So you take an image and then you would, using the tool within the set sequence info step, click this measure button. And as you can see, what we're going to do is measure 1,000 microns, and this is 1 millimeter scale here. So we click measure, and you simply drag from one point on your calibration standard to the other, and it will calculate automatically the scaling for the particular video acquisition that you had. So once this step is actually completed, you can save that. You don't necessarily need to do this for every video. As long as your acquisition conditions remain the same, you can just utilize that scaling by entering it at this step in the workflow. So now that we have our scaling set, save that, we'll go on to our next step. So in our next step, what we're looking to do is actually define the threshold level for our image sequence. So our images are binalized and segmented based on this threshold value to identify potential worm objects. You can do this manually by adjusting the slider. As you can see here, we are adjusting the slider and we're getting good representation of the worm. Alternatively, if you've set your scaling already, you can just click this auto detect button. WormLab will scan through the image and automatically optimize the threshold level for tracking purposes. There are other tools here in this image adjustment step. We're going to go over those a little later. In this particular video, we happen to have good contrast and relatively even illumination here, so we don't have to do too much with image processing. So once we've set our threshold level, we go on to the next step, which is the detect step, detect and track. So detected worms are described by a geometric model that's based on the center line of the worm and its boundary. We basically use these detection parameters here on the right to define which objects we're actually interested in tracking. So you can see here we have windows set up for area length width to length ratio. And in order for those to be accurate, all you need to do is actually click on a sample worm. So you click on a sample worm here. As you can see, we've created the center line, and this is the geometric model, essentially the center line and the boundary lines for the worm itself. And we've automatically filled in these detection parameters based on the sample that we picked. So now if we were to click the detect worms function, WormLab would sweep through the image, look for any objects corresponding to this detection parameter setup. As you can see, it detected 4 worms within these parameter setup here, but it did not detect this worm here on the right. It did not detect that worm because that one was a little bit smaller, so it doesn't fit into those parameters. So this is a method that you can actually use to discern say, tracking of adult worms versus juvenile worms that happen to be on the same plate. You can use this feature. However, if you wanted to also track worms that are a bit smaller, we just need to adjust these detection parameters. And you guessed it, easy way to adjust them is simply click on that smaller worm. You'll see these minimum values change. And if we now say detect worms, it detects all 5 worms in here corresponding to the new detection parameter windows that we set up. All right, so once we've actually set up our detection parameters, we'll go to the next step, which is actually tracking. All right, so tracking uses a deformable model estimation algorithm that fits that geometric model from the previous frame to the current frame. So on a frame-by-frame basis, we're going to apply this deformable model. Now, in the event that there's frames where there's some type of interaction, say, starting here at frame 245, this is where some worm trackers run into some difficulty and will stop tracking. Actually, this is where WormLab now has some unique advantages utilizing this multiple hypothesis tracking algorithm, which applies a set of possible worm locations across time, generating, you can think of it like as a decision tree structure. Entanglements and this or self-overlaps between worms can be properly resolved by finding the best fit across frames. That is, which branch of this decision tree actually matches the actual worm path. So it does this both forward, and we actually use this concept of backtracking to try to properly track through these more complex movements using this multiple hypothesis tracking algorithm. So the tracking step here really is just a matter of setting up what modes you want to use. So WormLab can track both crawling worms or swimming worms, depending on your assay. In this case, we're going to choose the crawling mode setup. And I will just decrease some of my multiple hypothesis tracking parameters here to make it go a little faster. And we'll start the tracking. So as you can see here, we're going frame by frame through the video and applying that tracking algorithm as we go. Here we're tracking through the entanglement. No problem there. And upon completion of the tracking, you'll notice that we now have actually assigned our head and tail appropriately for the entirety of the video. We can scroll through it now and see that. Tracking through the entanglements. And once you've actually tracked the video, you can now go to the next step of the workflow, which is analyzing the data. So here's really where the power of WormLab comes through. We have a lot of built-in analyses that you can utilize. But we do make it very easy for you to take the raw data that we've acquired through the tracking and easily export it for any of your own custom analyses. So for example, let's say you wanted all the position data for these 5 worms. It's very easy to simply export that position data with a click of a button, say export, and here, it generated on my other screen. Here's all the data associated with the position information of those 5 worm tracks. So our analysis is really meant to be very user-friendly and powerful as well as comprehensive, but also flexible, where you can export it, the raw data for your own types of custom analyses. Just to give you an idea of what this analysis window looks like as far as its features. On the left-hand side, we have the a tree structure showing the different types of analyses. As you can see, I'll expand these out. We have quite a few, ranging from position and speed through body shape morphometrics, bending angles, looking at body bends or head foraging type movements. We have another advanced analysis called label analysis. I'll go over that a little bit later. We have dedicated swimming analyses for anybody interested in more detailed swimming specific metrics. And then we do have some chemotaxis type analyses as well. So on the left, you'll choose your parameter that you're interested in. Let's say you're interested in moving average speed. On the right, you'll see the tabular data here at the top. And if you click any of the columns, you can see a graphical representation on the bottom here of the plotted data from the table on top. If you select multiple columns, you can look at multiple plots of data here for in this case, the moving average speed. In the bottom left-hand corner, this is where you actually can set up your user configurable parameters. So depending on the analysis that you're looking at, you may want to change the averaging window. So this is called a moving average speed. So there's an averaging window that shifts as you are, as it's doing the average analysis. And you can choose the number of frames that you would like to average across. So for example, we have here 15 frames. If I wanted to change that, say, to 10 frames. This is good for eliminating like outliers and noise that may have been part of the tracking process. You want to smooth out some of those by averaging across a certain number of frames. So let's say I change average across 10 frames. I say refresh data, and the system will automatically recalculate those values based on your new frame. Many people are interested, say, in our track summary. This track summary keeps growing based on customer feedback. But here you have a single point of information that gives you details on morphometrics associated with the worm, track length, track speed, wavelength, amplitude, body bends, distance and time traveled forward, distance and time traveled in reverse, and so forth. So we are constantly expanding these types of analyses and are definitely interested in any feedback you might have of what would work best for you. We also show things like reversals and omega bends, and give you kind of like a mobility snapshot here in the track summary. Popular analysis is also the this idea of bending angles. So you can look at bending angles again in the bottom left-hand corner. You can set up a like a body bending type report. What is your minimum amplitude threshold to be considered a bend? How long does that bend need to actually take place in order for it to be considered valid? Once you've set up your parameters, you can then go to this report tab, and it'll tell you how many body bends actually were implemented, or how many body bends were calculated during that time frame within the video. Let's see. So there's a lot here with analyses. I'm not going to go too far into this just yet, but I did want to point out this particular window so you can see what is available. The one other option I think that people could benefit from, let's say we're looking at the position metric here. If we have this option, it is by default on Kalman smoothing. Right here, we're looking at the raw data. So if we look at the raw data I'll zoom in on this graphical representation. You can see there are some jitters here. This is primarily due to phenomenon referred to as video jitter, namely changes in thresholding that will influence the overall data associated with calculation of, say, the midpoint in this case. So by using this common smoothing, what we do is actually smooth out those that effect, that noise effect generated by the video jitter, and we get a much better accurate representation of the movement of the worm. So I'm going to enable common smoothing, so refresh data, and we can take a look at that again. And now you can see this is much more representative of what our worm was actually doing during that point in the video, eliminating that video jitter effect. So these tools are all built right into the analysis to help you get the most accurate data possible. You still have your raw data by unchecking that box, refresh, still here, and then easily go back to your smooth data. All right. So an important consideration with respect to obtaining optimal results with WormLab is the quality of the video sequences to be analyzed. Ideally, we'd like to acquire videos with sufficient spatial and temporal resolution. And what I mean by that is preferably somewhere between, say, 10 to 12 pixels representing the width of the worm, as we see here. And I'll just turn these off so you can get a better view of that. So you can say here, with this particular video, we're getting about maybe 10 pixels across the width of the worm. And ultimately, we want an acquisition frame rate anywhere from, say, 5 to 10 frames per second for crawling worms and maybe 40 to 30 frames per second for swimming type assays. Since the detection algorithms rely on image segmentation based on thresholding of this binarized image, we really want to have good contrast between the foreground, that is the worm in this case, and the background. Yeah, not all videos are high resolution and high contrast with uniform resolution like we see in this sample video generated by the WormLab imaging system. But the good news is WormLab does have the flexibility to deal with less than optimal videos as well. So we're going to go into now that we've kind of an understanding of the general overview of WormLab, is to take a look at some of the parameter and video optimization tools that are built within the software that'll help us work with, say, less than ideal type circumstances. All right, so we'll open up a couple of examples here. So the first one we'll start out with here. I'll load in a video and I'll zoom out a little bit. So this video is about 45 millimeters in diameter. And as you can see, there is a illumination, brighter illumination area here. We'll call this a hot spot in the center, and it darkens towards the corners. Not that bad, but it could cause some problems with the thresholding as we take a look at this. And I just wanted to show you, I'll zoom in. As you can see here, these worms are definitely a lower resolution because we're so far zoomed out, but we should still be able to track these worms well because we do have some good contrast between the foreground and the background. So if we were to go through our steps that we've reviewed earlier, importing the image sequence we've done already, set sequence info, well, I'll just make a guess here that one of these worms is about 1 millimeter, just so we get something rough approximation. And I'll save that as our scaling. Then we go to adjust image. So here's where, normally we would take a look at the threshold and try to get a good representation of these worms by adjusting the threshold level. So if I adjust that out, there we go. You See, we have a good representation of the worm body there. I'm going to zoom out here. Oh, now the uneven illumination, it becomes a little bit more apparent. So as you can see, these darkened areas in the corner, once I've set my thresholding level so that I can properly detect the worms, these green pixels out here are not worms, obviously, but they will do the job of camouflaging our worms if they decide to go anywhere near those areas. So what we want to try to do is try to eliminate those extraneous pixels that WormLab would have to deal with from a tracking perspective. So the way we're going to do that in this particular video, since we have a relatively centered bright spot that uniformly is darkens as we go to the edges, our hotspot illumination correction tool is designed specifically for this type of illumination artifact. So if I click hotspot illumination correction here on the image adjustment tab, click apply, you can see we can really clean that up quite a bit, which is great. The other thing that we might want to take a look at here is the fact that there was this mark on the plate, and if the worm happens to go in this area, it could cause some problems as well. So maybe we'll use a little background smoothing to clean up the background a little bit if we can. There we go. So now we have good representation of the worm here, even though this is a lower resolution video. But we have a nice even background. So this is going to put us back into that ideal situation for tracking. So simply using a little hotspot illumination correction, some background smoothing, we can now achieve a good result as far as tracking is concerned. So this video is quite a bit longer. This is over 8,000 frames. Let's see, roughly 16 minutes long. So I'm not going to track this one in its entirety right now, but we can take a quick look at it. I happen to have done this already, so I'll bring it up. So I'm loading in the tracked project here. And now what we're seeing are the results of the tracking. The green represents the geometric worm model, and the red is the actual worm track. And you can see here, even through more complex movements, even at this lower resolution, for example, we just had a little reversal there, and now another small reversal followed by a mega turn. Even with this lower resolution, provided you've set up your tracking parameters properly and adjusted the video so you can optimize it for tracking, you can track through these more complex movements very accurately. But that's the key, to essentially get the video in a state that is going to work well for tracking, namely sufficient temporal resolution. Here we have 8 frames per second, which is good for crawling. Good contrast, even illumination. So provided those factors are in play here, we should get some good results. All right, let's take a look at another example. All right, so here we are, our first swim video. This is, let's see, a little over 2,200 frames, and over a 5-minute period. And the first thing you'll notice is the frame rate here. The acquisition frame rate is about 7.5 frames per second. It's not something I would typically recommend for swimming assays. As I said, we probably want to be closer to the anywhere from like 15 to 30 frames per second range. just so that we have sufficient temporal resolution, because as you see, I'll play this video. These worms definitely are going to move much more quickly compared to the crawling worms. So when you think about what's going on with our tracking algorithm, I'll just zoom in here so you can see, we're basically looking at, on a frame-by-frame basis, the change in the midpoint position of the worm. And if that position, based on the geometrical model that we've described, changes outside the allowed parameters for our tracking algorithm, we basically will then assign this worm a different ID. So effectively, you will not have a continuous worm track. Whereas if you have a higher temporal resolution, the change from frame to frame is going to be less, and we will be able to create a more continuous worm track. So temporal resolution is important. Here we have pretty good contrast. If we were to adjust our threshold, you can see just due to some of the unevenness of illumination combined with the shadow of the liquid itself, we have some areas that would potentially cause trouble for tracking. So we want to clean some of that up. And let's see, we can do that maybe with a little background smoothing like we did before. So I'll just apply some there, clean that up nicely. Knowing that we don't have the temporal resolution that we really need, what I am also going to suggest in a case like this under our tracking parameters is to change our position tolerance. So under this advanced tab, we have a couple of parameters that influence the algorithm, namely this position tolerance and shape tolerance. In a case where we have lower temporal resolution, we really want to try to help create more continuous worm tracks by increasing our position tolerance, namely how much of a deviation from one frame to the next would be allowed and still be able to call it the same worm. So this is a percentage of the body length. It's right now set to 0.3, so 30% of the body length. I'm going to maybe bump that up to say like 0.6, just to give us a better chance of being able to actually track those worms in a more continuous manner. Shape tolerance usually doesn't need to really be adjusted. I see some instances, particularly with swimming assays, where you may need to adjust shape tolerance. I didn't notice it too much here. If there's quite a bit of liquid media and the worm has the ability to move in the Z-plane. WormLab is a 2D tracker. So if we're moving in the Z-plane, it could potentially cause it to, let's say, look shorter or longer. Maybe you want to adjust your shape tolerance there. But the better solution is actually use less liquid media so that you really don't have that freedom of movement in the Z-axis. So we have it here to try to help in certain circumstances, but I would say normally you don't need to adjust that parameter. But let's go with our position tolerance just because we know we're at a lower frame rate here. If we detect our worm, let's see, we'll show our overlays, okay. And then we say detect worms. All right, they're all detected. I say, all right, let's choose swimming as far as our tracking mode is concerned. And let's start tracking. All right, I'm seeing worms kind of flashing in and out, and sometimes they're being caught, sometimes they're not. That's going to, I'm just going to stop this here. That's going to cause some problems. So given the adjustment of position tolerance, I don't really want to adjust it too much more than that. because then as worms get closer together, you can have problems with worm identity switching. So you want to be relatively conservative with adjusting those parameters. But what I am going to do is try to give the software a little bit of a better chance of making more continuous worm tracks. I'm going to take advantage of this detection frequency parameter that's in our detection tab here. So what this was originally designed for was a worm coming into the field of view. Essentially, we're going to apply the detection algorithm where we assign that geometrical model. We sweep through the entirety of the image. In this case, by default, every 10 frames, looking for new worms that are potentially entering the field of view. So what we could do is if we change this to 1, so now we're basically going to do this sweep every frame. What that's going to do is work in conjunction with that deformable estimation model algorithm that we mentioned earlier that's used for tracking. And it will give the combined algorithms together the ability to construct potentially more continuous worm tracks, even though we have a lower temporal resolution. So given that circumstance, let's try tracking again. All right, and now we see here that these worms look like they are actually tracking properly. So this is what we want. Excellent. So here we had a combination of issues going on. We had illumination issues, we had temporal resolution issues. By adjusting our position tolerance, changing our, which is here under the advanced tracking tab, by changing the detection frequency under the advanced tab of our detection tab, we were able to create more continuous worm tracks. So I will show the result here. So that we can see, here we'll turn on the worm tracks so you can sort of see how this works. So now we have continuous worm tracks, even though we were a little low on the temporal resolution standpoint. So the best fix would be to change the acquisition frame rate, increase that to around 15 to 30 frames per second. But we can still actually make this work with WormLab by adjustment of some of these parameters. And let's take a look at one more. Showing something slightly different. All right, again, another swimming assay. This one acquired at 30 frames per second and roughly 2 minutes long. Here we have 2 separate wells. So what we're going to do is go through our normal steps like we normally would. We can set sequence info. Again, I'll just approximate this by measuring a worm, just so we have some value there. Now we go to our adjust image stage where we're going to set our threshold. All right, so maybe go a little bit more. Now, as you can see, those wells cast shadows, and we have a little uneven illumination on the right-hand side of the image. So we definitely want to clean this up a bit and try to eliminate as much of this background as possible and clean it up as best we can. So what we can do is apply some maybe background smoothing here. All right. That looks better for sure. So we have good representation of the worm. This video has good resolution, you know, roughly, I would say maybe 6 to 8 pixels representing the width of the worm. Good resolution, good contrast, but we have all these extraneous type potential worm objects shown here in these green pixels that could cause some problems. So if I were to go to our next step here, which is the detection step, and say, all right, let's try to assign our parameters. So we've set up our detection parameters, sweep through the entirety of the image. We are picking up, you know, most of the worms actually pretty well here. We're seeing some extra objects out here, so that we really don't want. So one thing that we can do is try to focus where we're actually going to be doing our tracking. Now, some of you might ask, this is actually a good point, why didn't it detect these worms here? Well, the reason for that is if we take a look at what the software is actually seeing, I'm just going to turn on the, highlight those green pixels again, you'll see that this object, because they're currently self-overlap or overlapped on top of each other, this object actually falls outside our detection parameters that we have set up. So, you know, what I've seen some people do is click on this object so that it will pick up those worms. Well, that's not the suggested course of action, because by clicking on that object, you've now changed your detection parameters, and now you're looking at objects that correspond to this compound object, which is not what we want. The better way to deal with this is just leave it alone. When those worms separate, that detection frequency that we talked about, that happens every 10 frames by default, is going to pick up those worms and start tracking them. And then when we do our backtracking, it will actually come back and resolve that initial entanglement. So just a point there, do not click on compound type objects when setting up your detection parameters, because it will effectively corrupt them. But getting back to my point here, we want to eliminate some of the areas that we don't necessarily want to track within. And the way we can do that is by using this feature that we have. It's known as, so I just click this label editor tool. And what I'm going to do is I'm going to use this tool, and in this case, this contour tool. So we have different options here for different types of labels, circles, rectangles, the whole image, or in this case, a freehand contour. And I'm going to draw a contour that represents the area that I'm interested in actually tracking. I'm just going to turn this on maybe, so we can see a little bit better. So what I'm doing is just outlining an area that I can then tell the software to restrict to a particular region. So in this case, I'll just do this real quick. So I've created this contour, and when I go to my tracking tab, I can say restrict tracking to this particular labeled region. So it is actually not going to look at any data in this image outside of this delineated region of interest. So if we go back to our detection step for a moment here, I'll delete the worms. If I say run the detection again, now it's only going to use this contoured area. I say detect worms, and you'll see that we don't have any extraneous objects outside that region. So this is a way that you can use some of the image processing tools to clean up the background. But then if you can't clean up the background completely, you can now delineate regions of interest within your field of view to limit your tracking so that you can get accurate tracking results here. So if we were to start tracking, just so you can see how this works. And now, once this reaches 10 frames where these worms are not actually interacting with each other, we should start to see them potentially be picked up, provided the tracking parameters are valid. It looks like those worms aren't going to separate anytime soon. All right, so I'm just going to stop the tracking there, just so that you can see how this does operate, though. All right, and we can look at the final result. Let's see. Maybe we'll turn on some of those worm tracks so you can see that. Yeah. And we'll play our video. So yeah, as you can see here, we have a lot of body bending going on, a lot of thrashing, and WormLab will report all of this information for you. Imagine if you, the human observer, had to keep track and count of all these body bends for your thrashing assay. It's just not feasible in a realistic manner. So we will stop that there. So it's just a few examples of some of the parameter and video optimization techniques that can be used to deal with special circumstances associated with videos. Again, the WormLab imaging system is designed to really acquire optimal videos right out of the box. But WormLab is available as a software-only solution too. So if you have your own acquisition system, we can, try to give you some guidance on what would work best from an acquisition perspective, and then optimize parameters to work with your particular acquisition conditions. So what I'd like to now do is switch over to our other workflow known as our batch workflow. So this is very simple, but it is a way for you to become more efficient with your worm tracking assays and analysis. So as you saw, we went through this process of setting up a project file for tracking and then actually doing the tracking. Well, rather than do that in a serial sequential manner, WormLab has the ability to be able to queue up a series of project files. So you go through those steps of setting up your parameters, cleaning up your background, just save the file. You don't actually track. Once you have that project file saved, you can then use this batch tracking workflow to simply queue up those project files. And we give you a little hint right here, exactly what to do step by step. Set up your project files and then come down here to our file selection tool, click Add, and this will open up a browser window. You can now browse to where you may have some projects stored. I'm just going to pick a couple of projects here, and it will queue them up. So you can see this is their this is the path associated with them and the status. Then you just click Start, and it will actually start tracking each one of these videos in order. And you can do this, set this up before you leave the lab for the evening, come back the next morning, and you'll have all your tracking data already on disk saved for you, and you can go through the various analyses that you're looking at. So this is a nice way to efficiently track multiple projects overnight, unattended. Essentially, you don't need to be involved. So the reason why we did it like this was because, as you can see, different videos sometimes require different tracking parameters. But there are circumstances where the experimental acquisition conditions are very well controlled, so that you have a series of videos that all have the same tracking parameters associated with them. So we've recently added the ability to add videos themselves as opposed to project files. So you can just choose all your videos, load them up. Now what you do is you can pick a previously saved configuration file, which contains your tracking parameters, and it will apply this configuration file to all the videos. So what's really nice about this is you could set up this batch tracking queue very rapidly. Just put your videos in the queue. You already know what your parameter set that is valid for all those videos is going to be. You click apply and start, and now you can walk away, come back next morning, and you have all your data. So this was based on some customer feedback. Actually, at one of the international worm meetings, where we met with some customers, and they said, we understand why it works with project files, but we'd like it to work like this. So we're always looking for that feedback from you, how we can make WormLab more usable for the worm community. And this is just another example of that. So batch tracking is really just a way of taking what we've reviewed in the beginning of the tracking, using the tracking workflow, and just applying it to multiple projects or multiple videos. I will mention this applied configuration just because some people may not be familiar with that. We have this concept of a configuration manager. So I just click here and it opens up this tool. What you can do with this configuration manager is, here, I'll load in a video here. Okay. So what you can do with this configuration manager tool is, however you've set up a video, these parameters are now in memory. You can actually choose to save those parameters by clicking the save button here. Opens a window. You can name them. This will be, I'll call it weld plate. All right. And now I can recall those tracking parameters at any point in the future for, say, my batch processing. We've recently added this ability to not only save your tracking parameters, but if you choose this option, labels, click save. Hopefully, you can now save and recall parameter sets, as well as the associated labels with that particular setup. So this makes it really flexible so that you can apply this setup configuration to multiple projects all at once. All right, so again, batch processing is really a way to optimize your tracking capability. What I want to do now is take a look at some of the analyses here. We'll do that. All right. So here's a previously tracked video. This one, about 15 frames per second. It's about a minute long. As you can see, multiple worms. And let's see, right. There we go. Just so you can see it a little bit clearer. What I'd like to show you here is some of the analyses that you can actually, some of the more advanced analyses that you can actually utilize here with WormLab after you've done your tracking. So I'm going to click our Analyze Data window, and there it is. Okay. Click Analyze Data, and again, we can look at all the position information and so forth. Or in this case, moving average speed for the various worms. But what I want to show you here is a new type of analysis that allows you to dig a little deeper into the built-in metrics that we've already kind of reviewed. So, for example, we have this idea of label analysis. So we've looked at labels as a way to restrict tracking to a particular area, but they also could be used for the purposes of an analysis function here. So for example, let's say I wanted to just move our analysis window over for a moment, use my label tool, and I am going to choose the rectangle tool here. Make this a little bit bigger so everybody can see it. And I'm going to draw a rectangle there, and I'll draw another rectangle here. So we've already tracked this video. I just want to ask different questions, essentially. So WormLab is already telling me what my moving average speed of say, the worms are. I can pull that out, no problem. But what if I wanted to compare, let's say, the moving average speed of worms on the left side of the video here in label 0 versus the right side of the video in label 1? Well, using this label analysis, choose moving average speed, that's exactly what I can do. I can look at populations of worms using this region of interest tool and dig down into my data a little bit and ask different questions very easily. So although you can export data out and do this manually, possibly in other programs, we try to give you a very easy way to graphically utilize the label function as a way to interrogate the data a little differently. So this is an example of a what I'll call a spatial label. So we're looking at different areas. So you can imagine maybe having multiple rectangles representing a gradient that you might have throughout your field of view. That would be one possibility where you could use something like this. Another option, I'll delete those labels. I could use this full image label and click there. And right now, this label, as you can see here, is defined as the entirety of the image. And it's going from frame 1 to 915, all the frames in the video. I'm going to roughly say go to half. So end that label at frame 451. And I'll apply changes. And then I'm going to say, all right, let's apply another label from starting at frame 451 to end frame 915. Great. Now, what if I wanted to look at, say, the moving average speed of those two labels? I refresh my data. And what we now have is what I'll refer to as a temporal label analysis, in that we can look at the moving average speed of worms during the first half of the video versus the second half of the video. Or you can chop that up into whatever time groups you actually want. But the idea is you track the entirety of the video, you have all the information. What do you now want to pull out of all that information? And using this label analysis tool, you can look at your data a little differently and interrogate it and ask different questions that may be pertinent to your research. So that's one more, say, advanced type of analysis that I wanted to review. Another option that some people are not necessarily aware of, and I'll close this here, is this option for group analysis. So group analysis is, you can think of it as a subset of our standard analysis tool. It is growing based on customer feedback. So again, if you have any specific analyses that you're looking for that may benefit either our standard analysis or our group analysis, please let us know. But to give you an idea of how this would work, again, it's similar in design to our standard analysis in that we have our tree here on the left, our area in the upper area here for the tabular data, our graphical area here. What we're going to do is, using this add button for our group analysis, for previously tracked projects, we're going to choose multiple projects that are already tracked. I'm going to add them to our queue here. And what we're going to see is that we present you with various analyses for each individual project file. So this is project file 1, 2, 3, 4, and then an aggregate of all of these project files combined together. So looking at them as one group, essentially. And you can see these are the box plots associated with, this happens to be speed. We can look at moving average speed if we like. So you'll get a snapshot immediately of the aggregate of all the data that of your individual project files here. But we'll go one step further. Once you've added your project files, you can then define which project files belong to one group. Maybe group 1, that's going to be our control group, and we can define projects 3 and 4 as part of group 2, our experimental. Now that we have those defined that way, we can say, okay, refresh data and plot. So now we have basically our 4 individual project files, and then we have 2 group aggregate. One which is representing project file 1 and 2, and group 2, which is representing project file 3 and 4. So now you can immediately get a snapshot of multiple projects, multiple videos that you've acquired, group them together, and look for significant differences between those groups, and have a snapshot presentation right here through this group analysis tool. So those are a couple of some of the more advanced analyses tools that are available, namely the label analysis as well as the group analysis. As you can see, we offer many built-in analyses and metrics, plus you have full access to the raw data, as I mentioned previously, through the export function in the event you want to perform some type of custom work as well. We're continuously updating WormLab to provide you with a robust, easy-to-use worm tracking solution, but we do need your feedback. This is really a collaborative project for the worm community, and we do want to hear from you. With that, we'll now switch gears to the Q&A portion of the webinar. All right, well, Nick and I are ready to take some of your questions. It looks like Nick has been real busy answering some of the questions as they were coming in while I was giving the presentation. So let's see. Nick, why don't you introduce yourself and maybe we'll go over some of these questions.
Hi, my name is Nicolas Roussel. I wrote WormLab the beginning of it, and I'll be happy to answer the technical questions. I did my best answering the question I could answer. There was a lot, so I'm sorry I couldn't handle all the questions that came. Some of the questions Peter is better to answer them, so I left them for the end. Yes, I'm very happy to be here to help.
Great. All right. Let's see. So let's take a look at some of these.
Yeah. Actually,
All right.
I was reading a question about NIS element video format. Is it compatible with? And I had a quick look at the supported formats for NIS. So it says that NIS support AVI AP4, and so do we. So unless there is something proprietary in NIS, we should be able to support it as well. But feel free to download the demo version and try.
Absolutely. Yes. Uh, let's see. Let's see. Moving average speed bar graph shown here lack the error bars. Is that fixable? Yes, I think we should be able to make changes to the speed bar graphs. That shouldn't be an issue.
Honestly, I'm not ashamed to say that I'm not sure specifically what error bars is. If it's the error between the actual speeds and the moving average, we can provide a graphical display of this error. Yes, that is fixable.
All right. Let's see. We have another question here. Can we have multiple videos in one project file? The way the project file is set up, it is meant to actually only have a single video. And the primary reason for that is because we wanted to ensure that we were using optimal tracking parameters for each video. So to answer that question, multiple videos are not possible in a single project file. Do we have any tips for getting great videos with low background? Sure. I would say a WormLab imaging system works beautiful. But failing that, I would say you really want to look at the your illumination conditions. So we want a nice even illumination. We want to try to minimize, say, the amount of food or lawn that's on the plate. That will also generate better videos from a tracking perspective. You'll have less worm tracks. Let's see. And as far as the illumination itself is concerned, I would say utilizing collimated light tends to work best to develop a very high contrast image of the worm with an even illuminated background. Let's see. In the case of worm IDs getting relabeled, can you manually join IDs later on?
The answer is yes. Do you want to demonstrate it? It's in the repair tab. You just say, I want to connect track 1 and 7, and it will do that.
Right, yes. So there's a complete suite of repair tools built into WormLab to manually join tracks. And there's actually, in the latest version, release 2024, which will be available in a few days, actually, has an automatic join function built into it, which is something that will definitely improve the track repair timing. Let's see. Are you able to get frequency and amplitude of the oscillations of the worm's body? Sure. This is part of the bending analysis that is built into WormLab. And you'll be able to look at not only the amplitude and specify an amplitude for a bending report so that you can automatically calculate number of body bends given your user configurable amplitude metrics. Let's see. Can you please say something about the inner workings of the common filtering in the WormLab context?
Well, the common filterings, the inner workings is that of a common filter. The purpose of it is to clear the detection jitter because we can detect as accurately as the size of a pixel, and sometimes your detection is off by a pixel, and depending on the size of the worm, it can create, and the speed of the worm, it can create a jitter in the trajectory. So we use Kalman filter after the fact to smooth the trajectory and to get a better curve and better reading on speed. That's what we use Kalman filtering, and we do this after the fact. Kalman filtering is commonly used for tracking and is incorporated into the tracking. WormLab does not do that because worm behavior is not amenable to Kalman filtering. So it's only done for a post-processing purpose.
We have another question here. What is the best resolution and frame per second speed for capturing swimming behavior of worms? I would say we would want at least 15 to 30 frames per second, depending on how your worms are moving. That should provide enough temporal resolution. Can you have more frames per second? Sure, but then understand that that is going to increase your overall tracking time as well, because we need to track every single frame that's in the video. So if you were to use an unnecessarily high frame rate, that would just simply create longer tracking time without really giving you any additional information. So 15 to 30 frames per second works pretty well. We have WormLab old version. Kindly help to update it. Well, you can definitely get the latest versions of WormLab available from our download center. We do offer software upgrade support subscriptions, which allow you to access the latest versions of software. And as I mentioned, we have a new version coming out momentarily. So you should be able to take advantage of some of the latest features by downloading that latest version. Let's see. Can you send data? Can we send you data for interpretation purposes? We are always happy to work with our users, and we are constantly having a transfer of information back and forth, whether it be videos for parameter optimization or taking a look at analyses to see what will help you best answer your research questions. So absolutely, we want that dialogue with you. Can you take a video of can you take a single video of a 96 well plate, then analyze each well separately to statistically compare behaviors? What you can do, yes, is take an image of, say, a subset of those 96 wells, say like 12 wells in this example here, and then after you've analyzed the entirety of the video sequence, you could use that label function to then compare behaviors of the individual well. So you would use the circle function, circle label function, to define each well, and then you'll be able to compare the data between those wells. I think we're nearing what type of liquid are commonly used for swimming.
I'll save this answer for you.
Okay. Yeah. That's, yeah, N9 buffer. Yeah. I would say that that is quite common. How do the software parameters change when switching between crawling and swimming modes? Is it really the software parameters, or is it more the model that's actually utilized?
Well, they do not change much. They don't change at all, but you need to keep that into you need to keep that into consideration when you set your parameters. For example, swimming worms usually sees its center line move very rapidly, which means that you want to increase your position tolerance because the position of the worm will change very rapidly, much more rapidly than a crawling worm, because a crawling worm, the center points will move at the speed of crawling, which is much slower. So you want to increase your position tolerance. Shape tolerance should not be changed because the shape of the worm doesn't change, or marginally so during tracking. So depending on whether you're tracking for the purpose of tracking swimming worm or crawling worm, you need to adjust your tracking parameters and tolerance parameters. Now, if you have a fast enough frame rate, then what I said doesn't matter too much, because from one frame to the next, even when you're swimming, there is not much movement. So what I'm suggesting is something you need to do when your frame rate is relatively low and your worms are swimming fast.
Yeah, great, thanks. It looks like we're running a little short on time here, or we're running a little long and short on time. So I do want to thank you for joining us today. If we didn't get a chance to get to your questions during the session, we'll be sure to circle back to you directly via email. If you're interested in learning any more about WormLab or other products, you could just drop us an email, contact us anytime at info@mbfbioscience.com. And yeah, please don't forget to fill out the short survey at the conclusion of this webinar. Your feedback is very important to us. For general questions, anything from imaging to worm analysis, multiphoton imaging, we'd like to remind you that we have a series of forums where users can communicate with each other as well as our MBF experts at forums.mbfbioscience.com. Well, again, thank you, and we look forward to seeing you next time. Take care.